depurination. We have 2 more to go. I lied. I think I said 4. Such a liar. Deamination. What do you think this is? D and amine group, right? So remove an amine. Okay. So deam. So this means you're removing an amine group. Okay. It seems to happen more often to cytosine. Cytosine typically is the target of this deamination event. So here's your friendly cytosine. Okay, and what happens under a deamination event is this amine group is removed and replaced with an oxygen. That's what we mean by deamination. And cytosine is really prone to it. Thymine doesn't even have an amine group. And guanine and adenine, again, this is something where my lack of chemical knowledge, I should probably check with Crystal, but they're the double range structures, and they just don't, the deamination doesn't tend to happen as often to them as it does to the cytosine. Okay, so we end up with instead of an amine group, we have an oxygen. What does this look like? Everyone has the basis memorized, right? See, that was sarcasm. Sarcasm. This is uracil. So when cytosine gets deaminated, all of a sudden now it looks like a nucleotide that's only found in RNA. Okay, so all of a sudden you end up with uracil. Okay. And again, DNA can fix that, or sorry, the DNA repair mechanisms can recognize that that's uracil and fix that. The real issue that we have with deamination is when our cytosine is methylated. Okay, because we talked about epigenetics, right? We talked about adding marks to our DNA and the fact that we can add methyl groups. And which base are they typically methylated? Out of the 4 bases, cytosine. Okay, typically it's C's that are methylated. So if you have, right? You have your cytosine up here. And it's methylated. It's typically going to be methylated. And then if this undergoes deamination where you remove the amine group, so this is methylated cytosine. So if methylated cytosine then gets deaminated, okay, we end up with our product here having an oxygen. Now what does it look like? See, do you have all these memorized? You know exactly. The base that has 2 oxygens hanging off of it in a methyl group. I just mentioned there's only one base that doesn't have an amine group. Thymine. Okay, so this is thymine. So when deamination of a methylated cytosine happened, this is deamination occurred. then all of a sudden your base looks like 9. So repair mechanisms can pretty easily see uracil and say, okay, that's wrong. But they can't necessarily tell that this isn't supposed to be a T to begin with. Okay. So if you had, now you should have in your chromosome, you should have DT now paired to a G, right? Because if once this was a C that was methylated, that was paired to a G, but now also you have a T paired to a G, which clearly is wrong, okay? And DNA repair mechanisms will see that and want to fix that, but they don't know what the correct strand is. So should they put in a C here, or should they put in an A here to fix the lesion, fix the error? So that's where, and we'll talk about ways that the cell has tried to deal with that, but you can kind of see where if you're faced with this error and you don't know what the original base was, that you need some additional mechanism to recognize what the original base was. Okay.
When it goes to uracil, does it lose the other double bonded oxygen?
Oh, no, I don't. Sorry. Why not? Yeah.
Why is this more likely to happen to cytosine deamination?
So I mentioned, so thymine doesn't even have an amine group, so it doesn't have the thymine. And I don't remember why it doesn't happen to adenine and guanine. I should ask one of our resident chemist experts like Jacqueline or Crystal or somebody, or you guys who are on OChem right now, you should ask. But the guanine and adenine are double rings, so I don't face any of those.
Well, it's just because it's easier to convert from cytosine to everything else. Like if you have a 5-methylated cytosine, you can convert that to thymine. You can convert your cytosine.
Just because of the
to
structure.
uracil.
I don't, but it has to do with the attack that can happen on the amine group, the weakness of that bond. I don't, yeah.
Well, I mean, it's because you have thymine, you wouldn't be able to get it from a cytosine. You have to convert it to that. And
Oh, so you mean that we see it more as a result because you can end up with thymine from a methylated cytosine?
That's the more bio answer.
Yeah, that is a more bio answer. I was trying to think of a chemical answer for
Yes.
stuff. Yeah, I'll have to get back to you on that one. But yeah, it could be more in terms of biology. It could just be that we see this error show up as a mutation because converting cytosine is easy to make it, as Jason's saying, look like one of the other bases, where the other bases are so much more unique. If you take out the amine group and add in oxygen, it just looks like gibberish. You know, it looks like one of, or maybe a toddler. And then you have other, even larger, then we start to get dimers. When you have tautomers, a lot of times too, major chemical disruptions of the structure of DNA, right? You start to have bases on the same strand binding to each other instead of hydrogen bonding to their complementary base across the way. So then they're doing all kinds of crazy, and it'll be big three-dimensional disruptions of the structure. Yeah. But yeah, I'll have to come up with a better like chemistry, like hardcore chemistry answer. No, I'll have to get, as a biologist, I have to get a chemist to like actually talk to me, and that's hard. They're higher up on the food chain. And then physicists, they're the next, and mathematicians won't even, Okay. Last one, you can trust me this time. Last one. Polymers. Told you we would come back to them, so your little star in your note. But you have no idea why the star is there, but you're gonna indicate. So these are base analogs. So they look kind of like our discussion we just ended up had there about changes in the chemical composition of the atoms and the bonds. So this would be taking the bases and tweaking them. And they're isomers. They're considered isomers. They're around, they're available in your cell. concentrations of their atoms and or bonds are different from the normal bases, okay, that we consider being part of DNA. The problem in terms of a genetic standpoint is, and your book may or may not have pictures to show you guys kind of what some of these look like. So they somewhat resemble a normal base, but they're different. And the problem is that they can make abnormal hydrogen bonds. Okay, so they essentially, they're going to bond in strange ways. So like C to A and T to G. So because of the way the chemical structure is, they can actually make pairs that we don't normally see in DNA. So they can do those, what I just mentioned, the T to G pairing, or like C to A pairing. So then again, what happens is at replication, the cell doesn't quite know what is the correct structure. So which base should be inserted. So if you have an AC along your strand, DNA polymerase comes along at replication and isn't really sure which, should we make this into a GC pairing, or should we make this into, in terms of repairing it and replicating it correctly, should it be an AT or a CT pairing? Which one's the correct base and the original string? I just want you guys to know that there are tautomers that exist. They're isomers. They can look strange and they can make these odd hydrogen pairings. So A to C and T to G. Any questions? So these were all of our spontaneous. So this is stuff that can just happen inside your cell because of chemistry that's going on inside of the cell naturally, normally. Yeah.
So what about transposable elements? Or is that just
Transposable elements is another, would be kind of a spontaneous, and that falls under the, when we get to Fragile X, we'll talk a little bit, I can try to remember to talk about transposons then. That's when you're taking that small repeat and varying the copy number. So this was all kind of one base point mutation stuff. Yeah. So that's can cause mutations, but it falls more under that repeated unit category. And actually, we unfortunately again with I feel like you guys probably hear the same message in every class you take, that there's so much information about genetics that we could cover, and there's some of it we dump, and we don't really go into transposable elements all that much in Bio 311, but it's some really neat stuff. Most of our transposons are silent, they don't go around anymore. But that's the jumpy gene, if you guys have ever heard that, you should have. Okay. Yeah, so there's tons of other kind of crazy mutations and things that can happen that we're not going through in this class. But maybe we'll get to it in my Molecular Genetics Advanced 400 series class in spring. So we'll see. Lots of cool stuff. So induced, your category of induced non-spontaneous. We're going to take spring molecular genetics. Everyone's like, never again.
No.
Any class that says genetics. It's not going to be as problem solving based. I'm hoping to be more of a discussion based class, and we can go through articles and do more writing and stuff like that. Yeah. Talk about research.
That's going to become the seminar class?
Yeah.
Is that what you're saying?
This is a new course of 400 something. molecular genetics, special topics, yeah.
Show me
4 units, I think.
495.
495, yeah. 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 Okay. We're just gonna sit around and talk about hills and like, yeah. I'm making it sound all good. I feel like, oh, this is terrible. Maybe wait till you get your grade in this class. I can get wrong my face again.
The rest of the world.
3, 2. Giants are up. I see you're getting good mojo by being in class amount. Okay, so we can have chemical, and you can have physical. What's a mutagen? You know the word mutagen, assuming you guys. Something that causes mutations, okay? And whatever it is, it's just random class of something that causes mutations, okay? So chemical mutagens are the result of a chemical mutagen is going to be really similar to those spontaneous mutations we just talked about. Okay, so the result of it is you're changing the chemical structure. Depurination, deamination, any of those kinds of things. So this is going to look, in terms of the results, they're going to be similar to those spontaneous chemical mutations that we just talked about. That's all I want you to know for that. The only difference is you have an external versus an internal cause. Like some chemical. But again, oxidative damage, any of those could happen by exposure of oxygen. Physical mutagens, on the other hand, okay, this would be things like X-rays. Okay, this is why you wear that big heavy metal. X-rays, what they tend to do is they tend to create breaks in the backbone, so either single-stranded or double-stranded, but they tend to nick or break the backbone of DNA. They also often delete bases. Even oxidize a base. And then this whole nicking, I just mentioned it, but I didn't really. So this can be a single-stranded break, so SS break, or double-stranded DS break. And what happens when we have a double-stranded break? We've talked about some of the consequences of this. When we're talking about large chromosomal things happening. You know it, you just don't know the name.
You're talking about Monday's lecture?
Yeah. Like 2 days ago, 48 hours of memory just stuck into your head. Yeah, translocation. So this kind of stuff, if you have an X-ray, makes a double-stranded break. What happens is when you go to repair, all of a sudden you stick the wrong end of one chromosome onto the other chromosome. And then we end up with things like translocation. So large chromosomal rearrangements can occur just because of X-rays causing a double-stranded break. Yeah?
What do gamma rays do besides give superpowers?
Gamma rays and X-rays, so we could add that under our list, gamma rays and X-rays. And of course, you guys have probably had heard some of the stories of folks. So like Rosalind Franklin is the first one that comes to my mind, but there's other researchers. So she was one of the people that Watson and Crick stole information from to develop the structure of DNA, and she was doing X-ray crystallography, so she was exposed to tons of X-rays. Well, what do you think she died from? Tons of the X-rays, yeah. So this is before we knew anything about that. People were X-raying each other like crazy, and or just being exposed to it from work. And you guys have heard stories too of the radioactivity that people were exposed to, because we didn't know. Well, now that's why when you go to the doctor, you do scans or X-rays or things, they try to protect you as best as possible. And truly, I would be a conscientious patient in the sense of always make sure you ask whether procedures like that are necessary. I would be, pretty critical about exposing your body to these kind of mutagens. And some of them, the preventative care is much higher. You should go and get that X-ray. You should go and get, you know what I mean? Because that's a much stronger case to be made that that's a necessary and a far out change. But we do need to be careful about, let's see, one last physical mutagen. Running out of room. And this is outside. When you walk out there, what are you exposed to? Physical mutagens category. Dimers is what we call them. So we run around like dimers and indels, and we have UV damage to your cells. dimers being formed. So this is stuff that happens because of UV damage. And it happens throughout your lifespan, because we're all exposed to sunlight to some extent or another. And typically our body can repair this. It's just when it becomes overwhelmed, and we'll get more into that subject area about when these kinds of mutations that we've just described, when they really start to cause a problem, when the cell cannot repair it. Because it can repair a surprising amount of mutations and errors. Any questions about our physical mutagens? Yeah.
What was this last one called there?
Oh, sorry. Yeah, it's a 6,4. We're referring to the carbons involved, it's a 6,4 dimer. And the way I drew it here, this is a binding dimer. But it can happen with cytosine too, but we see it more frequently in binding. Oh, no, you lose one of these bonds though. Oh. Yeah, you lose one of the bonds between, yeah. You guys don't need to this is an OHIM, I won't make you show nucleophilic attacks and stuff, but, yeah. You don't need to. Go home, watch World Series, do whatever. I'll see you guys on Monday, I'll see some of you tomorrow.
I spent one.